More than 70% of bacteria are motile. They move along the roots of plants, they move on the soil, they move on the food we eat, on mucosal surfaces in our bodies, they swim in lakes and pond water. They're even moving in the scum behind the faucet of your sink. This motility is essential for the survival of some species of bacteria and for virulence in others. In this video, I'll describe some of the most commonly known structures related to movement in prokaryotes, but understand that at the recording of this video, there have been at least 2 more mechanisms for motility that have been reported in the literature. I'm not discussing them here because they're still under investigation, but the field of studying prokaryotic movement is alive and well. By the end of this video, you should be able to differentiate between prokaryotic flagella, pili, and axial filaments. You should also be able to describe the movement of prokaryotic cells and explain the significance of H antigens. As you'll recall, we were starting to look at all of the structures of prokaryotic cells. We were starting from the outside of the cell and working our way inwards. We started with the glycocalyx, and the next structure that we would see or encounter is this one, this long tail-like structure here, called the flagellum. Bacterial flagella, like the ones you see on these chlamydia cells, are tail-like structures that bacteria use for swimming. Each flagellum consists of a rigid helical filament, that's the structure in blue here, composed of a protein called flagellin. The filament is anchored to the bacterial cell wall via a protein hook and 2 or more basal bodies. The basal bodies are these kind of flat discs that are just inside and just below the cell wall. Basal bodies contain motor proteins, and when bacterial receptors detect certain chemicals or light, they send messages to the motor proteins in the basal bodies to tell them how to turn. Unlike eukaryotic flagella, which move in a whip-like or undulating movement, prokaryotic flagella are more rigid, and they rotate like the propellers on a motorboat. Typically, bacteria move towards chemicals called chemoattractants. Examples of chemoattractants might include things like nutrients or inducer molecules that help with biofilm formation. Chemicals that repel bacteria are called chemorepellents, and they can include toxic substances, harsh pH gradients, antibiotics, things like that. Together, these types of movement are called taxic movements or taxis. So together we're talking really about chemotaxis when we talk about chemicals. Movement towards light is called phototaxis. Prokaryotic flagella can have a number of arrangements around the cell. When you have one flagellum, the arrangement is called monotrichous. Mono, you'll recognize, means one, and the trick here in the middle refers to hairs, so it's like one hair. And this particular arrangement would be called polar because the flagellum is only located on one end of the cell. When there are 2 flagella and they're both located at the poles of the cell, that's called amphitrichous. Okay, so this would be amphitrichous and polar. When you have a tuft of flagella arising from the surface of a cell, then it's called lophotrichous. And when you have many individual flagella located all around the cell, the arrangement is called peritrichous. Peri means around, and then again, the trick here refers to hairs. So let's focus on bacteria. When bacteria want to move forward smoothly, they turn their flagella in a counterclockwise direction. This causes all of the flagella to tumble and fall behind the cell, forming a bundle. And as this bundle rotates, it propels the cell forward for about a second. When the cell needs to change direction, some of the flagella will switch to turning clockwise. This disrupts the bundle, and the cell stops moving forward as a result for a fraction of a second. The cell will reorient its direction in what is called a tumble, and then all of the flagella will synchronize again and turn counterclockwise, propelling the cell forward again. When the cell is moving forward smoothly, that's called a run, and when the bacterium stops and begins to reorient in space, like you see here, that's called a tumble. So bacterial movement consists of runs and tumbles. I've posted a video clip in your course space. Now would be a good time to pause this video and watch the link labeled Bacteria Swimming in your lecture videos folder for this chapter. After you've seen the clip, come back to this video and press play. How did you like that video by Paul Berg? Cool, right? Now you've seen one example of bacterial movement. Did you know that flagella are also antigens? Antigens are chemical sequences on the surface of microorganisms that stimulate the production of antibodies from B cells in the human body. Typically, antigens are part of a protein or a carbohydrate or a lipid on the surface of a microbe. We can use the reaction between antigens and antibodies to identify small genetic differences between cells of the same species. How do we do that? Well, let's look at the antibody first. This is a drawing of an immunoglobulin G antibody, or IgG. You can see that it's Y-shaped, and kind of the business end of the molecule is this part. So this is called the FV, or the variable region of the antibody. And there are thousands of antibodies in your body, maybe even more than that. But they all are distinguished by this FV region. This FV is designed to recognize different antigens, and it's determined by your genetics what type of antibodies you can make. When B cells that have these antibodies kind of embedded in them bind bacterium with the right antigen, then that B cell becomes stimulated to make more copies of the antibody that bound the antigen. So when it comes to bacterial strains, what happens is the FV region, which is represented by blue here and red here, it can bind antigens that are on the surface of bacteria. So in this example, we have two bacteria that are both of the same species called 1. So here's species 1, here's species 1, but they're slightly different genetically, and that genetic difference is shown in the fact that they have different shapes on their surface. So these are the antigens, and then the FV regions for different antibodies have the particular shape and chemistry to recognize those specific antigens. That's what we're talking about here. So strains that are identified by antigen-antibody reactions with flagellin will have an H in their name. They're called H antigens. So for example, there are lots of different E. coli strains. They're still all E. coli, it's just they're slightly different genetically. So one type of E. coli that's responsible for a lot of food poisoning cases is called E. coli O157H7. The O157 refers to one type of antigen. The H7 refers to flagellin as an antigen. So we can use this to distinguish this particular bacterium from others. Spirochetes use axial filaments for motility. An axial filament is a bundle of fibrils called endoflagella that are attached at one pole of the cell and wrap along the length of the cell. So if you look at this cross section, you can see endoflagella here. They're the little red dots, and you can see how they're kind of bundled together. So that's going to wrap around the cell. As the endoflagella rotate around the cell, They slide under the plasma membrane, causing friction, and this gives you the characteristic corkscrew shape that we see in spirochetes, and it causes movement forward. Cells like Treponema pallidum, which causes syphilis, and Borrelia burgdorferi, which causes Lyme disease, use this method of motility. Now would be a good time to pause this video and then go to the lecture videos folder in your course space to see how spirochetes swim. Fimbriae are short hair-like structures that some bacteria make on their surface for attachment. They help bacteria stick to surfaces. So here you can see these little pink hairs. These are the fimbriae. And cells can make a few to several hundred of them. We oftentimes see that bacteria will produce these in order to form biofilms. An example of a bacterium that makes lots of fimbriae for attachment is Neisseria gonorrhoeae. It helps the bacterium actually stick to the tissues of the urinary tract. Neisseria gonorrhoeae aren't the only bacteria that make fimbriae. E. coli cells will also use them to help stick to tissues. In fact, the pili that E. coli make have been linked to virulence. They help the cells stick to the upper intestinal tissues, and there they can cause the production of diarrhea, bloating, gas, stomach cramping, and infection. Pili are longer structures than the fimbriae, but they are not flagella. So they can be as long as a flagellum or longer than a flagellum, but they're not flagella. They're made out of a protein called pilin, and bacterial cells usually make only one or two of them per cell. These pili can be used for attachment, but they're also used sometimes for transferring DNA from one cell, like this one, to another, like that one. And you can kind of see that the pili are much longer than the fimbriae. If you look really closely at this image, you can kind of see the fimbriae only coming out about that far from the cell, whereas the the pili are much longer. In any case, pili are used for attachment. They can transfer DNA from one cell to another, and they're also used for motility. The type of motility that we see with pili, it's called twitching motility. Now, twitching motility is also sometimes called grappling hook motility because the mechanism looks a bit like how a grappling hook works. So my first introduction to a grappling hook was actually through watching old Batman shows with Adam West. I'd watch these shows with my parents. So this Batman had a bat-like hook attached to a string, and he would swing it and then throw it up into the air so that it would catch on a ledge on the side of a building. And you can see he's kind of pulling on it right here to make sure that the grappling hook part, is actually latched onto the ledge. Then Batman and Robin would kind of pull themselves up the side of a building to get to the Joker or Catwoman or the Riddler or some other villain. So this is a grappling hook. It's this hook that you throw up to a ledge or up to a tree or a cliff or something, and then you pull yourself up. So bacteria do this by making a pilus. They have a protein inside of them called pilin protein, and they assemble the pilin protein by secreting it to their surface. And as they make the pilin subunits assemble here at the surface of the cell, it makes like a little stalk. So the bacterium keeps assembling these subunits, making a longer and longer pilus until it contacts a surface or another bacterium. Once it touches a surface or another bacterium, it breaks apart the pilin subunits on this end, and that brings the bacterium closer to the other end of the pilus. and so it moves in that direction. So this is twitching or grappling hook motility, and it uses a pilus. So this is the third type of motility, gliding motility, and it's really interesting because up until, I would say 5 years ago, we thought that gliding motility involved pili, that cells would make lots and lots of glycocalyx and then somehow use their pilus to move. We now know that this is not true. It appears that there's a new type of motor that bacterial cells can use for movement, and what happens is that these cells use little sticky molecules called adhesins to stick to a surface. Then they move their motors, and something about how the motors move with respect to a membrane that's attached to the cell actually propels the cell forward in a helical direction. So it's completely different than what we thought it was. Now, what's interesting about this is that this type of motility is related to virulence again. So when bacteria find a spot that is really conducive to their growth, they'll sometimes organize to form complex communities like biofilms, and this can help the microbes survive, but it can harm human health. There's a bacterium called Capnocytophaga gingivalis, and basically a group of researchers reported that these common oral bacteria can shape communities by gliding over surfaces and transporting other bacteria that can't move on their own. So these Capnophaga bacteria have been observed using the gliding motility to transport non-motile bacteria from one location to another in order to form oral biofilms like plaques, which is incredibly interesting. So in this video, I've differentiated between prokaryotic flagella, pili, and axial filaments. You'll want to remember what these structures do. Remember, prokaryotic flagella are used primarily for swimming, right? They can be long, they have different arrangements around the cell. They're going to be used for swimming. We also talked about pili. Remember, pili have three different functions. One is that twitching motility, which is why I talked about that in this video. But in addition, pili can be used to help prokaryotes stick to surfaces and other bacteria or archaea. Pili also are used for transferring DNA from one cell to another. We also talked about axial filaments. Remember, we see these only in spirochete. They basically rotate around the cell, propelling cells forward and backwards. We talked about the taxic movement, or the taxis, of prokaryotic cells. Remember, we said chemotaxis is when cells move towards or away from a chemical attractant or repellent. Phototaxis has to do with attraction to light. Remember that cells move and runs and tumbles. I don't really require you to know that counterclockwise is the way that cells rotate their flagella to run, and clockwise is for tumble. That's not so important, but I do hope that you'll remember the runs and tumbles as far as determining the direction that a bacterium will swim. Finally, I also talked about H antigens and how they can be used to distinguish bacteria that are in the same species but have slight genetic differences. So I hope this video was helpful. In the next video, we're going to talk about the cell wall. That is a crucial topic for anyone who goes through a microbiology class. I look forward to seeing you in that video.